And so I hope today to cover 2D, and if time goes really well, then even three-dimensional morphogen patterning. Okay, so first of all, I want to talk about how do you measure size. And I want to talk about a particular gradient, because when we talk about morphogen gradients, you probably all think of these tissues or embryos. It turns out that you don't need to actually even be a multicellular organism. And so this is my artistic impression of a pombi. So fission yeast cell, it is between 7 to 14 microns. So at 14 microns, it divides, makes two new pombi at 7 microns length. They grow to 14 microns and divide. And this is, I think this is around 2 microns. And so pombi actually divides at 14 microns plus or minus a micron, which is remarkably precise if you think about it. This tiny, tiny capsule, and it actually almost does look like a drug capsule, is able to read out its division axis with high fidelity. And the question is, how does that come about? And one way that it appears to do this, or at least was hypothesized as doing it, was that there is a kinase called POM1. And POM1 is basically there's a microtubule network inside, I'm not going to draw it, is basically pushed onto the membrane of the pombi at the poles. So you have an input here at each pole. It moves around the membrane, the outer surface of the cell, and it forms a gradient. Because as it moves around, certain amounts dissociate and go back inside, and so you can create a subcellular gradient. So if I draw, if you draw the profile of POM1, it looks like this. And this was used as a, okay, there's two things here. One is, so if you calculate the intrinsic fluctuations in this system, they are very high. Right? The system is highly sensitive because there's not many molecules. And also, you can have very highly varying rates of, you've got a very dynamic process which is trying to push out the POM1 at the tips, and this basically means you get large variation in the POM1 injection rate. And so, actually, some work that I did quite a few years ago was to think, well, how can you make a stable subcellular gradient? And one thing that we realized, actually, it was one of those nice moments where I really felt that theory had its value, is that we were working with our experimental collaborators, Fred Chang in the US, and we couldn't explain their data. And the way we could explain the data is if we had a fast and a slow pool of POM1 on the membrane. And the idea being that the slow pool would be some sort of clustered form of POM1, and the fast would be more like monomeric moving around. And so we actually basically messaged Fred and said, look, we can't explain the data that you have. Can you take a look at because normally they imaged the midplane so that you could see the membrane, the edge all the way around. But we asked, can you look at the top surface and tell us what you see? And they very excitedly came back a few days later, and what you actually see on the surface are big clusters, and then you see them shrink and disappear, and then you see new ones form, you see them coalesce. And so this gradient is not constructed of sort of monomeric molecules just moving across the surface, but in fact is produced via a clustering mechanism. And so we actually then thought, well, can this explain what's going on? So we could consider that we have a fast form of the POM1, which I'll denote as fast. So this diffuses with, and I'm just going to reduce it to a 1D model. We can solve it on the surface. It's not too difficult. And I'll just write it down then. I'll explain what S is in a second. So that's the fast form, and then we can have a slow form. sorry. I'm trying to use different Greek letters just to mix it up a bit. So you can essentially have the slow form, if it bumps, sorry, the fast form, if the fast form bumps into the slow, like a cluster, it basically gets subsumed within the cluster and makes it bigger. But the cluster can also sort of disaggregate, and that repairs the fast form. And so you could just have this simple thing. For those that really care, this equation does not conserve mass, but it's just an approximation. And then these two terms say that the forms can go, basically they can fall back into the membrane. What we then assume is, well, we take the slow form to be pretty stable. So it can give and take protein on the surface, but it doesn't fall in. And we never saw clusters falling into the inner cell. And what's the consequence of this? I'm not going to go through the solution, but the nice thing is that what you find is that if you have some production rate, and we generally assumed it actually got produced, it gets pumped in this fast form, but we could have some balance, we could have some proportion in the slow form. Then what this does is it buffers fluctuations in J, the pumping in rate at the end. Because if you pump in too much, what happens is that you then form bigger aggregates. We have ds is a lot less than df. So you have more in the slow form. The slow form moves more slowly. And so what happens is that you increase so your normal profile looks like this. If you have too much being pumped in, it decreases more rapidly down because more gets put into the slow form, which is diffusing slower. And if you underproduce, then more stays in the fast form. And so you stay at so that you end up with a position where the readout is relatively constant. And so this idea seemed to work. We actually did some FCS where we tried to do FCS across the surface like this, and then try to detect the different forms. And we think we could just about see it, but it was challenging. But the first point I want to make is A, that you can have subcellular gradients, and B, that it looks like in order to sort of regulate noise and variability, one way that they get around that is that the gradient molecule actually can cluster. And clustering is a way of sort of buffering some noise fluctuations. It comes at a cost in other ways, but I don't want to go into that. Yeah, no, that's fine. You can remove that. And in fact, you can do this. So this is not conserving mass. You can do it correctly where you do the full clustering dynamics. So you can have 1, 2, 3, 4, 5 enmers. And in fact, if you really care about that, you can see, it's my only single author paper. So there's a single author paper in PRE where I actually go through the proper derivation of this, where you don't have, you actually keep track of the specific clusters.
Sorry, before we I think the code said if people do not believe in it. I just looked up on Wiki.
I didn't get it the wrong way around. Yeah, sorry. It does change the meaning. So, it's fake news. Okay, thank you for checking that. Okay, so why do we even care then, right? Why does it produce this? Well, Paul Nurse and Sophie Martin came up with a clever idea, which was the following, we have these POM1 gradients coming from the two ends. But this cell is growing, it's getting bigger. So let's just keep the left-hand side fixed. And so we're now going to grow out our gradient. And so if we look at it a bit later, the profile itself doesn't change. If we assume it's independent of the length of the cell. So we still have this coming down, and then of course it comes up here, right? But the key point is that the concentration at the middle of the cell, so this is L, or L, right? So the concentration at L over 2 is decreasing as the cell grows. So basically, you have overlap, you're growing. As you grow, your overlap is decreasing, all right? And so if we actually just take the POM1 profile to be a simple exponential, then our concentration at any point is some constant times e to the minus x. It's got some decay length of lambda plus e to the minus L. right? So this is your gradient from the left. This is your gradient from the right. And the key thing is that this is not just L, it's T. And so you can then ask, well, what's the concentration at the center of the cell, which is L over T. And so you can just write that as 2A. So if you plot now L versus C of L over T, it decreases down. So you actually now have a way by using opposing gradients to actually measure size, because you can imagine, well, let's imagine, we don't have to imagine, it's real. POM1 is a repressor of a protein called CDR2. CDR2 promotes the cell cycle. So what happens is that you imagine that levels of CDR2, so I'm just going to call it D for the sake of simplicity. So this is dependent on, so we're going to model the POM1 as being an inhibitor. So this is a standard inhibitory Hill function. So when a concentration of POM1 is 0, then this is on, and when the concentration of POM1 is high, then this is low, and then we'll have some degradation. I should think of a new Greek letter. What should I take? I'll take eta. I'll try to think of a new Greek letter every time I do a degradation. No, that would just cause confusion. But the point is that you can now imagine what's the concentration here. And so let's assume, and actually it's a very fair assumption, that the biochemical reactions and diffusion are much faster than the rate at which the cell is growing. So at each specific length, you can assume that this is all in steady state. So now that D is basically going as something over, right? But this depends on L. And so what you actually end up with is, if I plot on this, D at L over 2, it goes. And so you end up with, at a specific distance, the CDR2 accumulating and initiating the downstream cell cycle and division. And so this way, you can use opposing gradients to actually generate a specific position. And so I think this is very nice. It's one of those unfortunate cases where it's not as simple. So this was, the original papers were from Paul Nurse and Sophie Martin's lab. So they were Nature in 2009, I think. It turns out that if you check more carefully, it's not so straightforward. Sophie and Fred Chang and Paul have all subsequently looked. And so this model isn't quite right. It's definitely more complicated, as often biology is. But I think it's a very neat way of trying to see how you could build a system which could potentially read out a specific gradient and give you cell length. And this also works in multicellular systems. So I just want to highlight the paper, really. There's a paper from mchale in 2006 where they actually show, so there's no question, was there? Okay, where they show that if you have opposing gradients which are antagonists against each other, and you have a readout, that readout can be then sensitive on the size of the system. And so this was an idea of how you can measure the size of a growing tissue. And actually, these sort of ideas are also, as already alluded to this morning, you can also have things like sonic hedgehog and BMP as opposing gradients in the neural tube. And in that case, you can get very interesting dynamics and readout. And so opposing gradients are very, very interesting. And so this is just one example. There's a lot more in the literature. So I guess James Briscoe and Anika are good places to go, and good labs to go and check who worked on this extensively. Yes. Yeah, so in multicellular systems, it's always, always very dangerous. I'm not aware of where it's the same one from both ends in multicellular systems. It's always different. And it's normally antagonistic. So they are basically competing for a gene. And in this paper, they outline sort of basic thermodynamic models where you could argue that that would generate a readout. So actually, so perhaps not maybe this paper, but there is I'll just put up their names, but Anika and James Briscoe, they wrote a review in 2015. I can't remember the journal, but where they actually went through a lot of these different models in multicellular systems. But the idea I just wanted to really get across here is that by having opposing gradients is actually a way of reading out system size, because your concentrations at the middle, in particular, are sensitive to how big your system is. And so it's a neat way of measuring size in biology. Why have I got? Yes. I also I put in an extra, I have slide 2A, because based on the previous lecture, one can also ask, well, okay, this is all well and good, right? You can tell me that I have a sonic hedgehog gradient and I have a BMP gradient, but even in this region here, we come back to what we discussed on Monday, which is that there's going to be noise, there's going to be variability. And it turns out that that is real. And so in the neural tube in zebrafish, if you look at this boundary here, what you see is you don't see, like, in Drosophila, what you see is one boundary, then another boundary, and then, something else comes in, like, and so on. You don't actually see that in this case. What you actually see is, you have one cell like this. So at the boundary itself, so you start getting mixing. So on this side, it's clearly all pink. On that side, it's all white. But in the middle, you have mixing. And then the question is, how does that resolve itself? And that's generally a pretty, it's an interesting open question, actually. There is some work from the lab of Sean Megerson, which actually shows that when these cells take on specific sulfates in the neural tube, they actually take on specific mechanical properties. So they have different cell adhesion, they have different different cell tension, and that, they argue, is a mechanism for sorting out the cells. And so that by mechanical interactions between these cells, you end up with a sharp boundary between them. And I think Frank has also worked on how such boundaries can be resolved by mechanical competition. And so this is actually the first hint towards where I want to go on Friday, that you can have all these morphogenetic signals, but if you've got a growing system, a tissue that has to make a specific size and a specific shape, then you have to think about mechanics. And actually what biology has done is not just have to think about it, but it uses it to its advantage. Because it uses the mechanics to resolve the errors in the genetic readout, which I think is kind of cool. But that generally is actually, I think, quite a poorly understood area, particularly in vertebrate systems, for how this happens. Okay, so that's the part one done. Okay, so next I want to focus on positive feedback. So how do you read out the gradient? And what I'm going to consider is a gene of interest where I'm just going to call my gene A. So it's produced at some rate. I have some morphogenetic input. So this is a function of the morphogen acting on this gene. Okay. So the usual sort of response. So you have some production rate. It depends on the morphogen. There's no morphogen you don't produce. We could potentially have feedback. So, and we'll talk about that in a second. So if n equals 0, there's no feedback. As n gets bigger, there's feedback. And then we have degradation. And I will revert to my favorite mu at this point. And does this make sense? Well, it does make sense, but I'll give you the example in a minute. I'm just going to keep with the maths first, and then I will give you the biology in a second. But before we do that, let's let's just do our favorite trick. I don't like parameters, so I'm going to rewrite my concentrations in terms of a dimensionless concentration and my time in terms of a dimensionless time. And I leave it as an exercise to the reader to show that equals, so. Where alpha equals JF. Right? So we had 3 parameters there. We can reduce it down to an equation with 1. So, and that is important. And you've noticed that this one parameter is a tunable parameter. It is tuned by the morphogen as an input. Now, as ever, we'll be physicists, and so we're kind of going to go. We'll assume it's all in steady state. And actually, no, the first thing the physicist does is make a quadratic approximation. The second thing is assume it's in steady state. So what's the solution to this equation? Give me the easy one. Yes. So you stick A equals 0 in, you can get, I'll denote this AS for steady state. And then, of course, you're then left with an equation, A to the N. Now, for the mathematically inclined, if you want, you can prove to me that there's only 2 real solutions to this equation for general n, but we're not going to worry about that for the sanity of most of the other people. So let's just take n equals 2, and in that case, this reduces down to a quadratic equation, and you'll now see why I put in this factor of 2, because now my A is simply alpha m. Oh no, I won't write it that way. Alpha m plus or minus the square root of alpha squared minus 1. What happens if alpha is less than 1? I have complex solutions. Can I have a complex amount of protein? No. So, yeah, biology, it's one of my favorite things when I teach this in other courses, when I get told that there's minus 17 proteins in my system, and trying to explain to students that you can't have a negative number of proteins. But so you can see here that what this means is that we have a switch. If alpha of m is less than 1, then the only real solution is A equals 0. If alpha m is greater than 1, then we can have 3 solutions. Sorry. Okay. So what does this look like? So what we can do, actually, it's always helpful to plot things. So if we actually plot A, and then we plot dA by dTau. So we're just going to plot this function. So we know it's 0 at 0. Now, if we take n equals 2, right? When A is small, you have an A squared term on the first term and an A on the second. The linear term will win for small, so it's a negative, so you know you go down. As A gets bigger, then the first term starts to catch up and you go up. But then the first term saturates at high n, and so, but the last term keeps getting bigger, so you go down. So what you actually end up with, this is in this regime here, 1, 2, 3 solutions in steady state. Now, I understand you learned, or at least the people who were here last week did stability analysis, and there's some nodding. I'm going to give you the baby hand-wavy stability analysis. So, question, you did this, did you? Okay, then I don't have to. Which is the stable solutions? Is this a stable solution to this equation? It's mostly not, yes. Is this stable? No. This stable? Yes. So for those that don't know what we're talking about, what you can do on this is ask, well, what happens if I'm not at the steady state? What's going to happen? And the easiest one is, well, let's say I started out here. If I start out here, then my A is very large. I'm going to start decreasing in my number because I'm going to have massive degradation. So I'm going to go backwards towards here. And then when I get to here, the production and the degradation will balance out. The other easy one is if I'm very close to zero and I have a little kick, well, I don't really produce much because it's a little kick squared is tiny minus a little kick. So I'm going to go backwards. So you can work out which direction you're going to go in at these 2 points. And then it takes a little bit of thought, but you can see that it goes like this. So if you think about the way that you would move along these lines, so this is basically like you're playing football on the top of a very big hill. So if you kick the ball off the top of the hill, it rolls down. Whereas in this case, you're playing football in the valley. So even if you kick it up the side of the valley, it will roll back down to you. And so you're stable. I'm not allowed to cross. That's a problem. OK, I will have to migrate back. So whilst I'm rubbing up the board, try to think about where I'm going with this. What do you think is going to be the kinetics of this system? I guess, in a way, but it isn't a point of no return. You can return, and I'll show you in a second. But the key thing is that if we, so we can think about what's the consequence. So what we can do is if we plot, so remember we have this formula down here for what we're looking at is this formula. And we have these 2 terms, so I'm doing it in steady state. We have this degradation term and a production term, right? So it's useful to just plot those 2 terms. The degradation term is easy. This is the just proportional to A. And then we have the production term. So the production term is some hill function, right? Now, if our morphogen is low, so we typically, you can take f of m. Let's take f of m here to be something like m to the 4. You can take some reasonable function. So if our m is small, so we have a small alpha, then actually if we plot this, the first term, it looks like this. Oh, that's right, I don't want the j. So we have one solution. Our system is off. It doesn't matter. It might have a small amount of morphogen, but it's not sufficient to push it in. If we now ramp up the amount of morphogen so that we have a higher response, then you can see our free solutions. And so this is now a stable on solution. And so what you actually get if you plot M against A is that at low M, it's off, it's off, it's off, it's off, it's off. Switch is on. And I'm using the word switch deliberately because it actually does behave like a switch. Essentially, the intermediate level concentration is a transient state. It's not stable. And so you literally do jump from being off to being on. And of course, one can quite legitimately ask here, well, is that at all realistic? And I'm very pleased that Munir mentioned something relevant this morning, which is yes, it is. And so one way is a gene called Tin Man. Did I mention Tin Man yesterday or the day before? Didn't? I did complain about Drosophila naming convention. Can anyone guess what is the phenotype of a Tin Man mutant? If you know the answer, don't answer. Can you just, and I guess you have to, I'll give you a clue, which is the Wizard of Oz. It doesn't have a heart, yes. So for those of you that know the Wizard of Oz, it has the Tin Man who doesn't have a heart, and the Tin Man mutant does not have a heart. And Tin Man has got multiple enhancers. There's a Tin B, a tin D, and a tin C. There's also a tin A, but I don't remember what it does. But the important thing is that you've heard this morning all about twist. Twist is this important mesodermal factor that starts specification of the mesoderm, and it turns out that tin B is activated by twist. So at stage 8, 9 of development in the Drosophila embryo in the mesoderm, the mesodermal cells start expressing tin man under the driving of twist. But then at stage 10, something interesting happens. And I don't believe this is understood to be known why, but for some reason, there is a switch in the enhancer driving expression of tin man. It goes from being tin B to being tin D. So this is some epigenetic change, something probably to do with the chromatin accessibility, but I don't believe the details are currently known. Tin D is a very cool enhancer because it depends on the binding of tin man. And it also, and nicely from the lecture before, it depends on DPP. DPP is expressed from the surrounding tissues, forms a gradient across your mesoderm, and so essentially what you get is that only those cells with high DPP and which are expressing tin man already can then stay as tin man positive. All other cells do not. So that means that all the other cells in the mesoderm that are not expressing tin man, that are far from DPP, lose tin man expression. And cells which are not in the mesoderm, even though they might have DPP as an input, do not express tin man because it's required to be, it auto-activates itself. And then actually, once that's stably put, it goes on to tin C, and the regulatory elements of tin C are, again, not well understood. But this is how you can have a nice simple system where you can go from this initial mesodermal factor you heard about this morning that drives the initial specification, but then you use the morphogen not to define the initial field, but to refine an existing field into a specific layer of cells. And on Friday, I will talk for about half the lecture about the heart, because that's actually what half my lab does. And so I will show you images of what the heart looks like and how this happens. But I wanted to emphasize here that this is really not implausible. And if you go and look at the tin-D enhancer, you can find nice tin man binding domains. And if you mutate them out, then you lose the patterning. And so this appears to actually be real. But is this where you only ever get positive feedback in biology? And the answer is most definitely not. And so I want to slightly shockingly jump away from genes and regulation, which is what has been largely the last 3 days, just to talk about something else, but it's exactly the same maths. I want to show some relevance. Who's heard of GTPases? One or two hands. So GTPases are crucial molecular regulators that regulate membrane, cytoskeleton, and a lot of other interactions, including endocytosis and so on. And how they are basically an intracellular process that's driving different parts. And so you get localization of GTPases in different points inside the cell. And it's been a long-standing question about how do you regulate that? And so what I'm going to talk about is one GTPase called RAB5, which is specifically related to endosomes and, well, other things as well. And what this does is it actually has two forms. So it has a GDP inactive form and a GTP active form. And this is controlled. Does anyone know what regulates the going between the GDP and GTP states? Fun? Yeah, I think I heard what I wanted, but you have these things called gaps and gaps, which regulate this. And so what it basically looks like is you have the GDP form in the cytosol is attached to something called, say, a GDI, which is a anchoring molecule in the cytoplasm. And then you've got your membrane down here. And when it's activated as a GTP, it's anchored to a membrane. So the anchoring is essential for its function. And so these two can switch between each other. So the GEF promotes switching of GDP to GTP, and the gap takes it off. And a question which has been proposed for a long time but never really demonstrated is, does the GTP bound form play some sort of role in regulating its own binding? So, i.e., is there positive feedback in this? So to answer this, I actually worked with Martin Lusser, who's at IST in Austria, and so he's an expert in vitro reconstitution. So the problem is that this has always been studied in cells. This is a crucial cell process. If you make mutants, then you damage the cell, and it's extremely complicated. Like, they affect an awful lot of things. And so what he did was, well, he purified the GDI, purified the GTP form of Rab5 and the GEF. And then and the key thing, which people have done this before, but what they hadn't done is done it with a supported lipid bilayer, because the binding of the GTP to the membrane is essential for its function, and so for its stability as well. And what they found was really quite remarkable, is that if you looked as a function of time, from the moment that they added the GEF, then they would see. And what they were doing was doing turf microscopy, which is basically a way of imaging only a very thin layer. And I'm not going to go into why turf does that. But essentially, you're just looking at the pool which is on the membrane, right? And so as soon as when you add the GEF, then you get a response. But what they found was that as they did, so this would say 80 nanomolar, and then 60, 30, and then so what they saw was a really sharp switching in this system. And what was also interesting was that it had a time delay. And in fact, if you plot the time delay versus 1 over the GEF concentration, it was almost exactly a straight line. So if you plot the GEF concentration versus the active, the fraction bound, so this is the GTP form. Basically, it's, and I think, if I remember rightly, this was at 28 nanomolar. And so you could actually, the beauty of this is, so there's no cell biology here. This is in a solution with a lipid bilayer where we know the exact concentrations of the 4 things that we have, which is just the GDI, which is the anchoring in the cytosol, the GTP form, and the GEF. And in fact, in this case, we have no gap. And so we could get a beautiful switching behavior, and I was like, well, okay, I can model that. Let me come along with my positive feedback equation. So the idea being here that essentially once the GTP binds with the GEF, and then essentially that makes it easier for the next GDP to come in, and the next, and the next. And so you kind of have a positive feedback loop where they keep binding. And so I was like, okay, this should be nice and straightforward. I worked on switches. This is a switch. But what I couldn't explain was this time delay. Because when I fit, I should leave on this equation. Just if you take that equation, that's what I thought, right? You can fit this one, say, like this, pretty well. What happens is when you try to fit this one, it looks like this. And then if you try to fit this one, so it's very hard to get it to actually stay low, 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 and then shoot up. And it turns out that you can't do it without doing naughty things like tweaking parameters and making them depend on the, you can do it, but you have to make these parameters also then depend on the Gef concentration, which I didn't want to do. And in fact, there is a much simpler solution to the problem. Anyone want to guess what the solution is to this? Yeah. This, I'll give you the clue. This is correct, or not correct. This is a very good representation of what is happening. But the way I solved it was wrong. What have you been hearing about from Munir? You've been hearing all about noise and variability. So here we have a small number of molecules. This is a stochastic process. It is wrong to solve it deterministically because at a local volume, you actually have large-scale differences in the numbers. So if I now go back and instead of solving this in a deterministic manner, but I solve it exactly the same equation, but I use a Gillespie solver. Did you talk about Gillespie? No. So I don't want to get into it, but you can solve it numerically on a computer. Basically, you just generate random numbers and decide which reaction takes place. And if you do that, then I could get beautiful curves like this. So this would be the stochastic, and green is deterministic. And so this is, I think, a beautiful example of an important point, which is that when we often think about noise, we think of it as a bad thing. You know, this stochasticity, this variability, isn't it terrible? In fact, it's not necessarily terrible. And indeed, stochasticity can sharpen switches, and it can create specific turning delay times and so on, which is induced by, essentially here it's because the time it takes for an event to happen is dependent on you getting the local concentration right. So this I'm plotting is the mean, of course. If I was to plot this data, what I got from the simulations is I had a curve here, I had a curve here, I had a curve here, I had a curve here. But on average, the mean was where the experiment was. Yeah. That's a good observation. Yeah. So I think yes, in a sense it does. Yeah. It does a bit similar, yes, because we can't, it was hard to make the deterministic solution sharp enough. So in fact, the stochasticity, A, gives you the time delay, and B, it sharpens. And that's a good point, thank you. Yeah, but I'll tell you how to do it. So you have to do a... Yeah, okay, so the technical answer is you can do Hill functions as propensity probabilities. It's a bit dirty and should be done with caution. This is not that nonlinear. So in that case, it works. When you start going to seriously nonlinear systems, you can't do that. And you have to break it down into the individual steps. So this is what I would call a phenomenological model, and it kind of works. I actually had a student who solved the full, I think it's 6 or 7 equation system, and you can solve that as well using Gillespie, and it still gives you the same answer, or very similar. But for the sake of today, I do not want to write out 6 equations. But actually, that's a good point. I'm giving you a phenomenological explanation. We actually have in the paper a full explanation. But the reason I didn't want to talk about Gillespie is because, although I'm hoping my note is here, where is page 6? That's not good. It's behind. Good, my fingers. So actually, at this point, I met up with one of my old supervisors, and I was telling him this story, and he was like, oh, that's very nice, but come on, you can do better than that. You don't have to use Gillespie. You can solve it properly. And I was like, I don't want to solve it properly. But you can, and actually, it turns out it's pretty straightforward. So the best named equation in science, one not to say when you're drunk, is the Fokker-Planck Equation, which tells you what the probability, the change in probability of a dynamic system is over time. And you can actually write it in a diffusive way. So this is now, it's a stochastic process. You cannot say X leads to Y at this point. You can only say X leads to a probability of this at this point. So this is now the change in a probability due to some input X. And you can write this as I hope I got this right. I haven't double checked my notes actually, but. And the idea here is you have an effective diffusion term and an effective degradation term. And you can actually rewrite this in terms of these. And so in fact, if you now, I used A here, I'm just going to switch to X just for consistency's sake, but you can write this as, and the important thing is that this is plus X, and then this is minus X. And this is actually based on, I should say it so, oh gosh, I should have checked the spelling of her name, Alexandra Valchek. So she's at Curie and Paris, and she wrote a very nice review exactly on this, yeah.
There's a slight trouble. If D depends on X, probably you should have one derivative inside.
Yeah.
Otherwise it's a bit...
Yeah, you're right, you're right.
So then it's d of x, derivative of p of x.
Yeah.
No.
Which way around is it? It's
d of x, p of
yes,
x.
yes.
The derivative of a current.
You're right. You're right. I knew I shouldn't have done this off the top of my head. Okay. This is, I never remember, it's, yeah. P of x, sorry, it's then P of x. Yes, P of x. Yeah. Is that right? Then you don't get the D2, D term. When you get the second derivative. Yeah, true. Okay. I need to check this. I'm not sure. I think in the notes it's that. I will go back and double check. I know what you're on about, because if the diffusion constant is dependent. I think this is the formalism that works in the Fokker-Planck case, where you're making an approximation for the diffusion term. But I need to double check. I will go and check. I know that I did it right when I did it in the paper, because you can do some simple checks, like if you set this to 0, so you don't actually have a positive feedback term, what type of motion do you recover? You get Brownian motion, okay? So you recover the Brownian motion and you can check your statistics all work correctly. But the point is you can solve this numerically. This is pretty straightforward to solve. And if you do, then you can actually find something quite interesting, which is if you draw a phase portrait of essentially, let's just say, the feedback. That's the A1 term, and then the basal term. So this is the A0. So this is like just random binding events which happen without the GEF, and then you have the feedback term. And what we want to do is we want to have a switch. We want the system to be on or off. And so we can define a switch as, for example, so in the steady state, if there was no feedback, it would just be x equals A0. So this essentially, if you're in the steady state with no feedback, you have 0. And if you are above that no feedback state, then you have some positive S, and you want S to be as large as possible. And if you actually work this out, then you find, first one to die today, you get a region here where S is large. That doesn't work. S is large, and here where S is small. Why is that? Well, if you have a high basal production rate, then your switch looks like this, because you're already virtually high on. The feedback just tweaks it up a little bit, so you don't really get much of a switch. So you have lots of stuff, but it's not having a switch-like behavior. Down here, you never switch on. There's not enough stochasticity to make the switch, whereas where you want to actually be, where it's highest is around here, where you have a low activation, low basal activation, and occasionally by stochastic noise, you would jump up far enough to actually switch on the system. And so then you get a very steep response. And it turns out that if you look at the phase space for the experiments, it actually looks remarkably similar. So the example I just wanted to make here is that this is the idea of positive feedback being important, and that stochastic positive feedback is also essential. We're not the first people to think of this. There's a famous paper from Altschuler and Wu where they looked at how CDC42 positive feedback determines the position of the bud in budding yeast. And so, and again, that was dependent on stochasticity. Because, well, the master equation, you can't, as soon as you cut this term in, you can't solve it. Right? And I've tried. Okay, there is actually a deterministic solution. There is a solution using the master equation, but it's ugly as hell. The Fokker-Planck is nice. Yes, you're absolutely right. It's an approximation in the high end limit. But the point is, it's actually simple, and I like simple. It keeps me going. And so my attitude is, I think the important point here is I wanted to understand the phenomenology of the system. And I think this approach reveals that quite neatly. The gory details we did in Gillespie simulations. So we actually did simulate the full system with the Gillespie algorithm, and it works very nicely. And you get slightly different, but pretty much the same, whereas this is relatively straightforward. No, you see it, well, I don't know what you call a small system. So you see it in heart formation, but you could say that the system itself is small because it's acting at the level of the DNA. But then you see it in single cells. Yeah. Yeah, exactly. Yeah, probably less of a role. So this is intracellular. So for anyone who wants to read more about this, so that's the name of the first author, and it's still on the bioarchive. We have not got in yet. But I will move on now. Okay, good. So next I want to talk about negative feedback. I'm not going to go into as much detail. So let's just imagine now we have a negative feedback system. So now we have dA by dt is equal to, again, we have our morphogenetic input, but now it's negative feedback, so we have the A0 on the top. So we have, again, if we plot the line proportional to A, how many solutions does this equation have in steady state? In fact, any value of n, any value of the how many solutions are there? If I actually wrote this as but in fact, I believe, I don't know if I can say n greater than 0. I'm definitely positive for n bigger than 1, it's true. How many solutions are there? Real solutions? 1, yeah. Always draw. If you draw, it's obvious. There's only one point that they cross. And so negative feedback systems basically give you one stable state. But it turns out, so this is something where you've got something in it. Inhibits itself. It's pretty boring compared to the positive feedback. Okay? Positive feedback, you had all this amazing behavior. But yet, this is an extremely common motif. It's a very common interaction that we see. Do people want to conjecture what's probably the main reason for that? There's more than one. One? Yes, oscillation. Let's say, I'd rather say fluctuations, because oscillations can be sort of deterministic in a sense. But it's a way of handling fluctuations. If if you move up, then it puts itself back down. If you go down, then you push yourself back up. And so what it means is you can get a very stable level of protein in your cell, or a very stable response to your morphogen. So if you want to have a specific response to a morphogen at a specific location, it's a pretty good way of doing it. So, and actually that contrasts with positive feedback. So I gave you all the cool stuff about positive feedback, but there's a negative side, which of course is that it amplifies noise. So whilst that noise can help the switching, if you want to be precise, it's bad, because all that noise gets amplified by positive feedback. But where do we see, and in fact you've seen from this morning's lecture, that you end up with a very common motif in development. So this is our morphogen, and we are going to promote two factors A and B, but they're going to inhibit each other. Right? So this is a very common idea. And if we consume that A responds to high levels of M and B responds to low levels of M, then if we plot, so our morphogen is coming across our field like this, and then A responds to high levels, and what have I used? Orange. Let's see. So this is B, and then this is A. But then these two repress each other, and so the negative feedback gives you nice behavior like this. And it turns out in, for example, the Drosophila embryo or in the neural tube, that these interactions are sufficient to actually drive specific boundaries. In the neural tube, it gets even more complicated, where in fact you have 3, so you can then have and you can get all sorts of interesting dynamics that go on here, which I'm not going to go into detail, but the point is this is how you can define single cell precise boundaries. Yeah, sorry. Sorry, what kind of? Well, the idea would simply be that A is a gene that has very low affinity binding sites for M. And so you only get production when M is very high. B would generally have high affinity binding sites, but it would have a lower production rate. So essentially the idea is that this would get produced everywhere, or this would get produced everywhere across a large field if you didn't have the A, right? And if you didn't have the B, then A would still only get produced in a narrow regime. But generally the idea is that this is produced more when it's produced, but its threshold for being produced is much higher. And so that rule then can make this system work. Yeah, so it varies, yes. So you can, in this case, it would, I can't remember which way the parameters are actually. There is a specific order to them, I would need to check. Again, the person I'm going to cop out on this one, partly because I want to get on. Go and look up James Briscoe's lab, and there are numerous publications over the last 10 years where they really develop this. And they have some beautiful theory papers as well, where they go through it in detail. But I want to kind of move on from negative feedback, because we've only got 10 minutes, but I want to make the point that negative feedback has two good things. One, it really reduces noise. And two, it's a good way of producing sharp boundaries between domains. The specifics of how these promoters and enhancers respond to that, I would almost leave to Munir and others to explain. They know far better than I do about what's happening at the genetic level. Okay, but let's now get on to something. So far, really, I've just been talking about simple motifs, positive feedback. But we could imagine a three-component system. So you can have feed forward, say, so A promotes B and promotes C, and C promotes B. Or you could have, so this is feed forward. Or you could have feedback. So you could have A promotes C, promotes B, promotes A. And so there are lots of ways you can do this. It turns out that for 3 nodes, I read this in Urielon's book, and I never sat down and proved it myself. There are 13 possible free-node patterns, which seems like a rather odd number, but I think it's reasonably fair. But the question then is, if I had a network of n nodes, so each one of these is a node, and let's say I have n nodes. It's interesting enough, how many of these triangular patterns of a particular type do I expect to find? And it turns out the answer is not as obvious as you'd think, because if you think of, I want to take, so to try to make a triangular network, imagine I have a node and I have two vertices coming off going to other nodes. Now, well, if I have a system of n nodes, well, each one can basically create something like this. So I'd expect to find roughly a number of, in my system of n nodes, I'd expect to find roughly n of these patterns. But I want to make a specific triangular node and motif. And so then I need a specific interaction to come to link up those other two. But the probability of me choosing one of those connections from a system of n is going as 1 over n, because I have n choices, and I got one choice to make. So in fact, the number of free component motifs in a system of size n goes as n to the 0. It's kind of weird. But there's an interesting observation from this, which is that this is not what you see in biology. You see specific triangular patterns coming out more often than not. And so that's very strong evidence that they're being selected. Because if it was a random network where you're randomly trying to make triangular nodes, you would actually not scale with system size. But if you're trying to make n of them, but it turns out that you do. You see specific nodes coming up more often than others. It turns out you get two types. I'm going to draw down, you can imagine multiple ones. So this is the classic feed-forward loop. It is very, very common, and you see it in many gene interaction networks. This is called coherent, because all the interactions are kind of helping each other along. You could also imagine this. That would also be coherent, because A downregulates B, which downregulates C, and A, which in effect upregulates C, because you're repressing the repressor of C, and you're also upregulating it directly. So this is still a coherent network. Any question?
Very selective.
Yeah. Yeah, that would be the separate. Yeah. Okay, yeah. No. Yeah. So I'm sure an evolutionary person would kill me. Yeah, you're absolutely right. They are more common than I would expect. Now, whether that means it's being selected is a different. You could hypothesize that that is because of selection, and I'm not sure how much it's been tested. But that is thank you. That is a very good point. No, well, but in this case, this is where I'm not the largest omics fan in the world, but omics can be very powerful. And this is where we have huge networks, and you just can unbiasedly sync with them, and you get these out. So yes, that can happen, but I don't think in this case that's the case. Can you wait about 5 minutes? I will tell you. But I wanted to say the other one. It turns out that there are two very common motifs, and the other one, and it was someone mentioned it yesterday, is incoherent. So in this case, A promotes B promotes C, but A represses C. And you might think, why the hell would you do that? You know, are you mad? And it turns out that it's not. There's a very, very good reason for it. And in fact, the answer was given yesterday. I will be interested to know if anyone remembers. So let's first of all talk about this first one. The first thing is, who here knows what Boolean algebra is? Yes, this rings a bell. Like, this is the idea of, like, you have gates, you have AND gates or gates. You have two inputs, and they have an operator. And so an AND gate is you need to have both on to have an on out. This is how you make an AND gate. C needs A and B. If you have A and B, you have C. If you don't have A or B, then you don't. Or you can make it an OR gate, because you just have to have on the enhancer binding sites for either B or A. So this is how you actually can make AND or OR gates in your cell. And what we can think about is, if we plot as time, let's assume that we start with everything off, and I need 3 colors. That's 2, that's 3. So we're going to take A to be white. So we're just, well, okay, A is going to be the most boring. So A is just at time point 0, we turn it on. Right? We just not worry about it. We just pump in A. So what happens? Well, let's take B to be, so this is A. Okay, what happens? And I take it as an AND gate logic. Now, B turns on, right? It starts to increase, and it will come up to some level. Now, of course, C, then when B gets above some level, above some threshold, we're just going to take the simple model, which is when it's above a threshold, it's on, and when it's below a threshold, it's off, right? And then we can imagine the same. So now C is off, off, off. We get to this point, and we turn on, right? Now, what happens? So if you see here, I have a time delay between turning on A and seeing C turn on. What happens if I turn off A? Immediately, thank you. So now I remove A, and immediately both B and C decay. So this is, if you're an engineer, you might have heard the term sign-sensitive delay element. You have a time delay for on, 0 time delay for off. What is anyone, like, I don't know if anyone's used them. Well, obviously they have used them. What's a device that you use every day, or maybe most days, which has this sort of logic, where it has immediate response, but a delayed. What? Not quite. It's your human brain. I'm more thinking, what happens if you're on the 4th floor of your residence? You use a lift, right? When you press the open button, the door opens immediately. Does it immediately shut behind you? No, it actually waits some time. Often if you're in Singapore, there's this joke in Singapore, they're always desperately pressing the close button as fast as they can. It doesn't make any difference, the door still closes slowly. But there's this, and that's because they're engineered that way, because you don't want the door to slam onto someone as they're trying to come in. So lift doors are actually engineered to exactly the same logic. They have it's the opposite way around. They have a quick on, open, and they have a slow off. And you can engineer both ways around. And so it turns out that these motifs are very common. And so although this is obviously an AND or an OR gate, there's actually more interesting stuff to it. And in fact, it has interesting temporal dynamics. And in my last few minutes, and that's quite good timing, I'm going to finish with the incoherent one. So I know that there's at least one person in the audience who knows the answer. So let's do the same again. We're going to ramp up A immediately. Now, what happens? So A promotes B and it represses C. So what color was B? B was blue. So B starts coming up. And then, no, have I got this the wrong way around? I've drawn this the wrong way around. Sorry. The other one was correct, but it's not showing what I want to show, so I will ignore. Everyone also does something else quite interesting. So in this case now, A is promoting both B and C. So both A and C come up immediately upon A coming up. But now, once B gets above some threshold, then B starts to repress C. And if I assume it's kind of an AND logic, so B off and A on, then this then comes back down. So what we've done is we've got a pulse generator. So, all those fancy oscillators and stuff we played with in physics class when trying to make pulses and so on, well, biology does it as well. And you do it through such a motif where you have the specific order of gene interactions. And then just another common motif which relates back to the Tin Man one is that you can often see not negative but positive. What have I done? It's getting late. So this is called a self-connected triad, I believe. And this is what gives you memory. Because essentially, one of them get switched on by something. In this case, in the case of the heart, this is Tin Man, and this is the initial input. It then drives dorsocross and pineal downstream factors, but those downstream factors then turn around and start maintaining Tin Man. So this is embedded within this Tin C enhancer down the end there, which is not well understood, but we know this basic motif is playing a role. And this basically gives you memory in the system because once it's on, it kind of reinforces itself and keeps itself going. Okay, so that is where I will end for today. That's kind of gone through now I got it. A lot of the fundamentals, I hope, of how you interpret gradients and how you read them out in reliable ways and how you can get lots of interesting behavior and how noise can be important. What I'm going to do on Friday is actually talk about, well, today I've covered quite a bit of my own research, but we'll talk about more about it. So we're going to talk about how you pattern butterfly wings, how you pattern your back muscles, and then we're going to talk about how the heart actually forms and gives its shape. And I think on that I will end. Questions? And of course there'll be a quiz. I like quizzes. No.